Alright, so in this video, I'm just going to be talking about transcription. Well, in this part of the video, if you've already watched transcription in prokaryotes or transcription in eukaryotes, then you can skip this part and then go to the timestamps below. So yeah, let's get started. So the general info for transcription is that unlike DNA replication where we make the whole DNA sequence again, transcription it only focuses on the gene. So, if you remember from your class or our terminology videos, the gene is only a specific part of the DNA. If this is your DNA, the gene will only be like just this specific sequence over here. Now, not both top strand or bottom strand is the one that is being transcribed. Usually it's just the if the top strand was the template, then this one would be the one that will be transcribed. If this was the template over here, then this one will be the one transcribed. It's only either one. But at the same time, it can also happen where both sides have a gene, but normally it doesn't. So if we're gonna summarize what a general info of transcription is, it's basically the process of making an RNA strand, because we can make any kind of RNA out there, which is like mRNA, tRNA, rRNA, miRNA. There's a lot of RNAs. So the main point here is we're just making an RNA strand using a gene as the template. And then in both prokaryotes and eukaryotes, there's a very general steps on how the transcription works. So if we're gonna make the general steps, I'm pretty sure this is something that you guys have heard before, at least in your other biology classes. First step is initiation. During initiation, this is where your assembly of the different proteins or enzymes and then the recruitment of the RNA polymerase. Second, elongation. Elongation is the part where RNA polymerase starts adding nucleotides from the 3' end. Because remember, we can only add on a 3' OH. Like only on the 3' end that we can add nucleotides. And then the last part of the general steps is termination. Both prokaryotes and eukaryotes have different types of termination, and I will talk about that in the second part of the video. So if we're gonna visualize this, let's say this is our DNA, right? This is our DNA. Let's say this is 5'. Now, let's just say this is the gene. This is already the gene. I'm just not gonna make it a DNA. Instead, I'm gonna make it a gene. And then the top part is the template. Then that means the bottom part is the coding strand. Now, during initiation, the strands will open up. I'm just gonna make them widely apart from each other as a sign of them opening up. It will be open up with using a helicase enzyme or the actual, sometimes RNA polymerase has the activity of helicase. And then the recruitment of the RNA polymerase itself. So let's say it was recruited around here. Because remember, we make from 5' to 3' always. Now, during elongation, RNA polymerase will just keep adding nucleotides. Right? Because remember, it's kind of like the same thing as DNA replication, where elongation starts from 5' to 3'. Same thing here. The RNA polymerase will be adding at the 3' end. This is the mRNA that is being made by the RNA polymerase. Now, last step is termination. Termination, basically, RNA polymerase will be kicked out, and then the mRNA or the RNA strand is disassociated with the template strand. So now you have the matured RNA strand. So yeah, that is the very general information about transcription. Now to the second part. Alright, so in this part of the video, I'm going to be talking about prokaryotic transcription. One thing to remember about prokaryotes is that they don't have any membrane-bound organelles. So that means they don't have a nucleus or endoplasmic reticulum, stuff like that. So everything happens in the cytoplasm for prokaryotes because they don't have any membrane-bound organism. So transcription happens in the cytoplasm. And then for prokaryotes, especially in bacteria, they only have one type of RNA polymerase that does the job of elongation and initiation mostly. Promoters are recognized by sigma factors. Normally, if you hear or see the word factors in biology, you can assume that it is a protein or a type of protein. And in this case, the type of protein are called sigma factors. And sigma is normally denoted as this. So sometimes you might see in the exam or some paper that they have something like this. That means it's a sigma. So the specific promoter that you've learned in class are between -35 and -10. This is the specific promoter region that you've learned in class. That is part of the promoter. And then the specific type of sigma factor is called sigma 70, or you can spell it out, sigma 70. So those are just very specific terms, but for generalizing it, the promoters are being recognized by sigma factors. Now, after transcription, there is no further modifications, just like how, if you remember in your class or in your lesson, eukaryotic transcription, they have some form of modifications like 5' capping or poly-A tailing or splicing. For prokaryotes like bacteria, they don't have further modifications of the mRNA. And now, termination can either be row independent or row dependent. There's two types of termination in bacteria. Alright, so before I go and try to visualize each one of them, RNA polymerase is a holoenzyme. Because if you remember in class, there's like these different enzymes or proteins that are like clustered together. Those are called a protein complex. And specifically, this type of protein complex is called a holoenzyme. Now, if we're gonna like generally define what a holoenzyme is, it's just a combination of different proteins to do the actual job. And in this case, to do the elongation part of the mRNA strand. And then one fun fact that I just want to share about RNA polymerase for bacteria. This is only for bacteria. Bacterial RNA polymerase has helicase activity. If you remember what helicase does, it can break hydrogen bonds between base pairs. Bacterial RNA polymerase can do that by itself. So think of this like transcription in bacteria, like literally RNA polymerase is doing everything else. It's unzipping the DNA as it elongates the strand. Alright, time for visualization. Alright, so during initiation, if you remember earlier, if you watched the first part of the video, you remember something like initiation, elongation, and termination. For initiation, remember, in bacterial or prokaryotic transcription, we have this promoter region called -35, -10. This can be found in the DNA sequence or in the gene sequence itself. Now, this promoter will be recognized by sigma 70. I'm gonna denote it as a square, so just to make it easy. Now, this sigma 70 will go ahead and bind, or just go to the promoter region of it. As you can see here, it bound to the promoter region. Now, this sigma 70 is gonna send signals out there to recruit RNA polymerase. Now, once RNA polymerase is recruited, sigma 70 is released while RNA polymerase is doing the elongation. And remember, this is an important part, RNA polymerase is adding at the free 3' OH always. Every time you see something that is being elongated, it's always adding at the free 3' OH. Even in DNA replication, it's the same thing. Now, after elongation, termination could either happen between row-dependent termination or row-independent termination. Let's go ahead and talk about row-dependent first because it's easier. Alright. So let's say during elongation, when the RNA polymerase is elongating the strand, there's a this is for row dependent, by the way, row dependent termination. So there's a sequence called RAT sequence. I just put it here like this is not a separate thing. It's just part of the mRNA, but I write it in red just to make it more specific. So in the mRNA strand, RNA polymerase will be transcribing this RAT sequence over here. Now this RAT sequence can be recognized by this row enzyme. I will make it a triangle just to make it easier to see also. Now this row enzyme will bind to that RAT sequence over here. And then from that RAT sequence, it will literally chase the RNA polymerase. So first, we have the RAT sequence that is transcribed. And then row enzyme will come in and bind to the RAT sequence. And then the row enzyme will chase from the RAT sequence to wherever RNA polymerase is elongating. So once RNA polymerase chases, once row enzyme chases RNA polymerase, like in this part, it will literally just tell RNA polymerase to stop elongation. Now, once it stops elongation, both RNA polymerase and row enzyme will just dissociate. And now we have the full mRNA strand. That's literally just row-dependent termination. That's why it's called row-dependent, because it depends on row to stop elongation. Alright, so next one. Row-independent. Now, row-independent is kind of hard to... And it's not really hard, but it's like might be confusing. So, let's say we got here, right? After elongation, RNA polymerase suddenly transcribe these sequences like GGC, CGG, like there's a rich sequence of G and Cs. Now, remember, G pairs with C, right? Within that RNA strand, those G and Cs might form a hairpin loop because it looks like a hairpin. Now, this, if you imagine this GGC, GCC, this is the RNA strand that was being made over here. Literally, those G and Cs will pair up with each other. So, like, you have this strand. Like, if you imagine without the actual sequence, it will literally look like this. That's what it will literally look like. because the G and the C's will be pairing up with each other. And then by the end, there's like a bunch of U's because that's how it is. And then because of this hairpin loop, it literally disrupts the RNA polymerase, making it dissociate. And then we have the terminate, like it literally just, after it dissociates, it's the termination of the elongation. So now we have the mRNA strand. One example I can give is like this. Imagine over here is the RNA polymerase, right? RNA polymerase is going and going and going, and then suddenly this loop over here forms, and you can see how RNA polymerase is like changing its direction or like being disrupted. So like at that point, RNA polymerase will be like, oh, you know what? Maybe I should stop. That's why it's dissociating. Or you can, if let's say this was at the end, RNA polymerase will just fall off because the hairpin loop is literally disrupting its direction. That's one way to imagine it. So yeah, this is the video for prokaryotic transcription.